Well, just as we had a special section about the actual process of mitosis, now we're going to go fairly quickly through the process of meiosis when we actually look at what happens to the chromosomes and how they operate during this process. So some of these images are already going to be familiar with you, as are some of the terms that go along with it. And as before, just as with mitosis, you're going to have to remember the terms, these different phases. They don't actually change between mitosis and meiosis. They are just illustrated in a way so that you understand which one of them is in mitosis and which is in meiosis. So the summary part here is that we produce haploid gametes. So this is where we transition from the diploid organism to the haploid gamete. And in order to do this, we need two divisions. The first one of these is called meiosis 1, and the second one is meiosis 2, and these are collectively known as meiosis. So far, so good. Now, one big important difference between mitosis and meiosis is that meiosis has errors, and when these errors occur, they are not actually a problem. In some ways, they're actually the coolest thing about this process because they allow for a certain amount of genetic variation to creep in without hurting anybody, but perhaps allowing something to get better, a process to get better, genetic material to just be exchanged, and so on. Now, interphase, as is shown here, is the same in both mitosis and meiosis. As you see, the chromatin is in the middle of that nucleus, the nucleus is surrounded by a nuclear envelope, and the chromosomes are undergoing replication. So the genetic material is doubled. This happened during mitosis, and it happens as a precursor step to meiosis. Then during meiosis 1, the homologous chromosomes separate. And I realize there is a little bit of a glitch. Somehow this writing on top here does not show up in your copies, and so you might want to write that in. This process here is meiosis 1, and it consists of 4 distinct phases. These are the same phases with the same names that you encountered in mitosis. The difference is that all of these are going to have a Roman numeral attached. This Roman numeral tells you that you're not in mitosis, but that you're in meiosis. It tells you more specifically that you're in the first part of meiosis. And so whenever one of these phases, like prophase, has a Roman numeral with it, then you know you're in meiosis. If it doesn't have a Roman numeral, then you're in mitosis. Now, prophase 1 is particularly interesting. And the reason why prophase 1 is particularly interesting is because of chromosome overlap. Some of these chromosomes, as is shown here and here and here, they actually overlap. Now, if you're a chromosome, and I'm going to draw one over here, so there is a chromosome, right? And chromosomes like this may have a long leg, so there's a long piece of DNA that's hanging out here. Well, clearly, if you have this long leg, it is possible that just by getting into close proximity of another one of those chromosomes, you end up with a little bit of hanky-panky going on. You have a little bit of a foot overlap, if you will. If you had chromosomes that had really short arms, let's say I'm drawing a different chromosome here, and it has a long arm on that side but a short arm here. These are never going to overlap. The longer legs, they can overlap, and in some cases they can even overlap twice, but the shorter ones don't. And so it actually depends on the chromosomes themselves and on what kinds of genes are on these chromosomes, on whether there's going to be any of this exchange or not. This process is called crossing over, because they're crossing over when really they're not supposed to or not expected to, but they do it anyway. And if that happens, then you may end up exchanging pieces. So the piece that is part of the blue chromosome becomes attached to the red chromosome, and vice versa. So it's an even exchange, which means the chromosome information, the genetic information on the chromosome, doesn't actually change. You're simply exchanging where the information came from. And so that is one of the things that makes meiosis very interesting, this genetic exchange. Now, it is possible that this genetic exchange doesn't work, and then you may not end up with any gametes. That's okay. But if it does work, sometimes it can produce new and interesting things, and this is one of the reasons why crossing over is so interesting and important. So these tetrads form, the tetrad simply being the two chromosomes lined up next to each other. Two of the same chromosomes lined up next to each other is the tetrad. And they're still composed each of two sister chromatids. The sister chromatid is one of these lines. That's one sister chromatid. Now when we move on with this, we go into metaphase 1. Metaphase was another one of those phases that we already saw. The difference between metaphase in mitosis and metaphase in meiosis is that here you have the pairs line up in the middle. The pairs are lining up in the middle. If you're looking at mitosis, the only thing that lines up in the middle is any old chromosome, any which way. They don't pair up. And so in meiosis, you find that the pairs, these tetrads, line up in the middle. And of course, the process remains the same. You have these spindle fibers attached to the chromosomes. The spindle fibers are the things by which the cell can catch these chromosomes and hold onto them in order to pull them apart. And pulling apart happens in anaphase 1. The sister chromatids during this time remain together. The things that are being split up are the homologous pairs. And that's why this is the reduction division, because you're starting out with 2 in each pair, and you end up with 1 of each pair in different cells. And so during telophase 1 and cytokinesis that goes with it, you end up with 2 haploid cells. And so with meiosis 1, the output, if you will, is 2 haploid cells, still with sister chromatids attached, and perhaps having gone through crossing over, so that there's a little bit of an exchange in the chromosome material. Now this then isn't the end of it, because now we follow both of these cells that we just produced into meiosis 2. And as I mentioned before, meiosis 2 is simply a mitotic division. And in this mitotic division, you have the same phases as before. You have prophase 2, metaphase 2, anaphase 2, and telophase 2 with cell division, cytokinesis completed. And here, these chromosomes act exactly the way they would in mitosis. They line up in the middle, the sister chromatids are separated, and then during telophase, the new cells form. So that's the second round of divisions in meiosis. Now, if we enumerated all the differences between the two, we would find that clearly, these are very similar processes, but meiosis has 2 steps, mitosis has only 1. Meiosis allows a certain amount of error to be introduced with this overlapping, whereas mitosis doesn't. So those are the kind of things that are different, but they're still very similar kinds of processes. So that's part 5.